All right, so today starts meiosis. So the big picture that we are going to talk about is what accounts for genetic variation in organisms. So genetic variation is the key to what we are talking about today. So why do you think siblings look different from each other? Say the second part of that again. Okay, so they get different genes from their parents. So one sibling might not get the same genes from their mom, okay, that one did, and same with their dad. So then why, we said siblings, so why do children look different from their parents? Great, so we're talking about sexual reproduction, so we have a combination of mom and dad. Okay, so that is why they look different from their parents, but a lot of times you might look more like one parent than you do another. Sometimes it's 50-50, it just depends, okay, on the genes and traits that we get from our parents. All right, so sexual reproduction involves the contribution of genetic material from two parents, and that is what actually results in genetic variation, okay? So sexual reproduction is two parents, right? So the offspring are what compared to the parent? They're not identical, so they are different, they are unique. So we have talked about this briefly when we did mitosis, and you did a little activity on this. You're going to do another one again because this is super important. So we talked about the terms diploid and haploid. So when we actually talk about a human, okay, human body cells contain 2 copies of each of 23 chromosomes. So altogether, we have 46. We get 23 from our mom and 23 from our dad. So we get a copy from each parent, okay? So each set of chromosomes is considered to be homologous. This is going to also take us into some terminology that we'll talk about when we get to genetics. So homologous, the word that you might want to pull out of there is homo. And homo means what? In one word. The same, okay? So they are the same size and therefore contain the same information. So this word right here, homologous chromosomes is going to be one of the key pieces to meiosis that we did not see in mitosis. Okay, so after we replicate in the S phase of interphase, those homologous chromosomes now form what is known as a tetrad. So when it makes a copy of itself, that is what is known as the tetrad. So you have, you'll see the individual, if I go back, okay? And then when we replicate that, duplicate that, that's when you get your tetrad. So these are some other terms that we've previously talked about: somatic cells and gametes. Okay, somatic cells are body cells, and this is super important. You should be very familiar with this term. So a lot of times you might not actually see body cells. You will see the actual biology term somatic cells. So these are all the cells in the body except for the gametes, which are our sex cells. So all the other cells in our body are somatic. So these are diploid. Our gametes, our egg and sperm, our sex cells, are what are haploid. Because when the egg and the sperm combine, we're getting 23 from each. So those are haploid, which in the past I have taught you means what? Half, okay? And diploid means the full amount. So that is always double. So haploid is N or 1N, and diploid is 2N. Okay, so somatic cells are what are made by mitosis. So we had talked about if we cut our finger, what happens when we cut our finger? Okay, so we lose blood, but eventually the skin will heal and repair, and then we grow and make new red blood cells. Okay, so that happens via mitosis. Now today what we're going to spend our time on is meiosis, and this is how gametes are made. So that is our sex cells, sperm and egg. So you're going to tell me what type of cell are egg and sperm? A gamete. What type of cell is in the skin? That's somatic, a body cell. What type of cell is a gamete? Is it diploid or haploid? Haploid. And how many chromosomes does it contain? 23. Perfect. What type of cell is a somatic? Diploid. So how many does it contain? How many? 46. Okay, so that's our full set. What kind of cell is made by mitosis? Somatic. Meiosis? Gametes. All right, so now we're going to get into meiosis. So meiosis, which is also known as the reduction division, is the process by which gametes are made. So it works, like I said, much like mitosis, but there are a few differences. Okay, so each cell divides 2 times instead of just once. So in mitosis, there's only 1 division. In meiosis, there are 2, because we are going to go through meiosis 1 and meiosis 2. Okay, so PMAT 1, PMAT 2. Next, each cell produces 4 daughter cells. How many did we make in mitosis? 2. Okay, so we only made 2. Now we're making 4 because it's going through 2 divisions. So we make 2 the first time, and then 2 a second time, so that is where we get our 4. Then, we have already gone over this, but the cells produced are haploid instead of diploid, because in mitosis, the cell is identical to the parent. In meiosis, it is different because it gets from the mom and the dad, so therefore, cells are haploid and diploid in mitosis. Last, each cell produced is different from the parent. So in mitosis, they are identical, and in meiosis, they are different. So we consider them to be unique. So there are a lot of ways in which meiosis can result in genetic variation. So I told you genetic variation is key for today. Okay, so what we mean by that is different combinations of DNA. So we're going to talk about a few of these. The first is crossing over. Okay, so during prophase 1, so remember I said we're going to go through PMAT 1 and PMAT 2. Okay, so during prophase 1 of meiosis, homologous chromosomes are going to pair up to form that tetrad. So we have homologous chromosomes. The chromatids are then going to cross over, like you see up here. We've got blue and purple, okay? And then the cross sections become exchanged. This word is super important. And crossing over is exactly what it says it is. You see the chromosomes, the chromatids, crossing over one another. So now this blue chromosome, okay, has a piece of the purple, and the purple has a piece of the blue. Okay, so this is one way that we can get new combinations of DNA, of genes. So crossing over happens in prophase 1. So we're essentially swapping DNA. The second is known as random or independent assortment. So when we say that something is random, what does that mean? Okay, you don't know what you're going to get, right? So chromosomes in this case are separated into gametes randomly. So it is by chance, not by choice. So that is why you inherit traits by chance, okay? Because you get a mix of traits from each parent. So you can't say, hey, I want this trait from my mom and this trait from my dad. Okay, it happens all by chance. Next is mutations. And we've already talked about mutations. And mutations are simply a change in the sequence of DNA. Okay? So sometimes there is a mistake and it is not caught. So remember, we talked about replication and we talked about how DNA polymerase comes back through and checks for error. Okay? It is meant to fix it, but sometimes that doesn't happen. And that's what results in a mutation. Four is sexual reproduction. Okay, so this is reproduction that requires two parents and it results in a variation because any combo of sperm and egg is possible. Okay, so when sperm and egg join, okay, this is known as fertilization. And when fertilization happens, this is when we get the correct number of chromosomes. Because the egg has 23 and the sperm has 23, when they combine, we now get our full set of chromosomes. Okay, so we take our sex cells, we combine them, and we get what is known as a zygote when they fertilize. Okay, so the sperm fertilizes the egg, and now we have a zygote with the full number of chromosomes. When we get into genetics, I'll mention it briefly today, but when we get into genetics, this does not always happen, okay? Talking about mutations and different things that happen, we don't always end up with the correct number of chromosomes. And those result in things like Down syndrome. And we'll talk about that more when we get into karyotyping. Okay, last, and this also gets into karyotyping, is non-disjunction. This is another big term. We need to make sure that we know this term and we understand this term. So non-disjunction happens when chromosomes don't separate correctly. And this happens during meiosis. So this leads to the wrong number of chromosomes. So instead of 46, someone can end up with 45, 47. Those are the biggest ones, okay? And it will result in genetic disorders like Down syndrome. And like I said, we'll talk more about this when we get into karyotyping and how to read a karyotype. Okay, so when we don't have the proper number, that is non-disjunction. So the chromosomes do not separate correctly. So up here, we see our chromosomes lined up. And when they go to separate, so this would be what phase? What metaphase? And then this would be anaphase. So when they go to separate in anaphase, if they do not separate correctly, like you see in the bottom, in the red, okay, that is what can lead to things like Down syndrome. So here is a picture of a karyotype. We will talk about these all by themselves, okay? And a karyotype is a picture of our chromosomes. So we have 23 pairs, the 23rd being our sex chromosomes. So this is also what tells us whether we are male or female. Females are what 2 letters? Anybody? What? Females are what?
X. Or X.
They're an X and an X, okay? Males are an X and a Y. So males are actually who determine the sex of a baby. Because the male can give an X chromosome or a Y chromosome, but the female always gives an X. Okay, so in this case, this karyotype shows us that this is a female because there are two X's. Okay, it also shows us the genetic disorder of Down syndrome. So someone with Down syndrome, also called trisomy 21, means that there is an extra chromosome on the 21st pair. Okay, so that is Down syndrome, and Down syndrome is the big one that you need to know. We're going to talk about a few others when we get to karyotypes, but Down syndrome is big. So Down syndrome, also known as trisomy 21, 3 chromosomes on the 21st pair. So that means instead of having 46, this child would have 47, because they would have an extra chromosome. So genetic variation is produced by meiosis, and that is what ensures that there is a wide variety of traits in offspring, okay? For example, the dogs, okay? So meiosis is what gives us genetic variation. All right, mitosis or meiosis produces 2 cells. Mitosis produces cells that are identical to the parent, genetically different from the parent. 4 cells. Chromosome number is the same as the parent. Half as many. Increases genetic diversity. Very good. Chromosome number is divided in half. Results in a clone. Mitosis. Crossing over happens. When they cross over, remember we had the blue and the purple chromosome. That's meiosis, okay? That's what helps give us genetic variation. Asexual reproduction makes a gamete. Part of sexual reproduction makes somatic cells. Mitosis occurs in reproductive organs. Which one? Meiosis, that's right. Somatic cells. Okay, and what does crossing over result in? Genetic variation, okay, or different combinations of genes. So up here just gives us a quick difference chart between mitosis and meiosis. So meiosis is sexual, mitosis asexual. In meiosis, we get how many from each parent? How many chromosomes from each parent? 23. So the total number is 46, but we get 23 from each parent. Whereas in asexual reproduction, it is the same as the parent. So if there's 46 in a blood cell, then it's going to make 46. Meiosis, 2 divisions. Mitosis, 1. Cells produced in meiosis is 4 because of the 2 divisions, and in mitosis is 2. Last, meiosis occurs in the sex cells, the gametes, and mitosis in the body cells, also known as the somatic cells. Does anybody have questions?